Alright guys, we're going to dive into proteins. This is topic 7.5 and hopefully you've been reading chapter 5 of your textbook because it's pretty foundational to understand those biomolecules as we go throughout this first year of IB biology. Proteins are pretty neat. There are actually a lot of proteins in your body, and we get the variations of proteins simply from around 20 to 23 amino acids. And so according to the order of those amino acids, the way that they're constructed and put together, we get a lot of different proteins. Proteins are also unique. We see them inside of our brain, and we call them prions. And so one example of a prion that has actually caused a lot of issues for people. Typically prions are normal, but sometimes they can be kind of wacky, and so we'll look more specifically at this in class. But there is an example of in New Guinea, there's a disease called the Kuru disease, and a long time ago in the 50s, cannibalism was actually banned because women and children mainly were getting sick and dying from this particular prion that went awry in the brain. So women and children were eating the brain of dead relatives because that was a part of their culture, and they were actually ingesting these prions, these proteins that went bad, and that was causing them to also die. And so recently we have found out that there is a gene that actually can protect a person from getting this disease, Kuru. And so back in the 1950s, we saw natural selection actually weed out people that don't have this genetic mutation, which is called G12-7V. And so, of course, through natural selection, most people, if you didn't have the mutation, you died. If you had the mutation, you lived. So obviously, over time, that's what we see more of in the population. And come to find out, we've actually learned from studying this tribe that this genetic mutation actually protects a person from Kuru. So the reason why I think that prions are interesting is that they're just ordinary proteins inside of your brain, but somehow they have become misfolded, and that's something that their scientists are trying to figure out how and why. It is very unique that there's a genetic mutation that can protect you from particular brain proteins. But the why do we get that misfolding in the protein? That's really interesting. And so if you remember back a little while ago, we had to kill a lot of cows in Europe because of mad cow disease. These cows actually had misshapen proteins in their brain, and the fear was that if you did eat the cow, that you would actually get these proteins inside of you and you would die. And it could have been a possibility because these prions can actually, the diseases that happen because of prions can actually cross over from mammal to mammal. So it was a good thing we did that. But we'll talk about more of that in class, and but now let's kind of talk more about the specifics of proteins and amino acids. So there are several different functions of proteins, but just to be able to name four, got examples for you. Proteins do a lot of things in your body. For example, one specifically called amylase. It is a digestive enzyme, and so it's something that can actually catalyze starch breakdown in your mouth. So the starch molecules will break down because of hydrolysis, and they will turn into glucose molecules, and this is something that's found in saliva. It happens immediately as soon as you eat something that's starchy. And another example of a function of a protein would be with hemoglobin. It carries oxygen from your lungs to the entire body. Another example that we've studied within year 2 material of IB is actin and myosin, and these two proteins work together to help your muscles contract. And then lastly, another example that's very common in our body is insulin. It is a protein, but it's a hormone that can help regulate the carbohydrate and fat metabolism inside your body. And so if you notice, you'll see that most proteins, they actually have a suffix of in. And if you see that suffix in, that's a really good indicator that what you are looking at is a protein. Proteins remind me of Legos. If you grew up with Legos, you may have had the really huge box that has 50,000 pieces of Legos, or nowadays you can purchase really nice kits. They have Star Wars kits, they have I don't know, probably Hello Kitty something. They just have a ton of things. Indiana Jones. And so the basic building blocks of proteins are actually called amino acids. And so I think of the individual amino acids as our pieces of Lego, right? They're all unique. They have different sizes and different colors. And depending on the order that you put them together in, you will end up with something that is awesome. This is the Sears Tower or the Willis Tower, but I refuse. It's the Sears Tower for my Chicago heart. But now they have these really cool kits that are called the Lego architecture, and you can make stuff from New York. You can make all these different skyscrapers. It's pretty nice, pretty streamlined, and looks really cool. An amino acid is made of three different main groups. You have an amino group, which is going to be the group on the left, and then you have a carboxyl group, which has a double bond with the oxygen attached to our center carbon right here in the pink. And then lastly, we have something called an R group. And the R group is considered a side chain. So every amino acid has the same amine group and the same carboxyl group. What differs for each amino acid is its R group, and that's where we can get specific properties for each amino acid. So our four different levels of our protein structure are the primary structure. This is literally a unique sequence of amino acids, as if you took every single amino acid, chained them together, you conducted a dehydration synthesis. You could see in this picture we have dehydration taking place so that we can basically form what is called the peptide bond. And we chain them all together, and just the sequencing of those amino acids is going to be the primary structure. Once you begin to chain those amino acids together, two amino acids are held together by a peptide bond, and we get what is known as a polypeptide. It is the string of amino acids. The secondary structure is if we took that primary structure, the chain of amino acids, and we actually saw foiling or coiling of that amino acid polypeptide. And so you'll have two different types of structures that we'll see. They're held together by hydrogen bonds. You've got the alpha helix and the beta pleated sheet. So an alpha helix spins sort of like the DNA helix, and then our beta pleated sheet kind of has this back and forth zigzaggy kind of structure. And right here you can see hydrogen bonds are holding the structure close together. Next we have the tertiary structure. From here it's a 3D folding of the secondary structure. So if I took a helix shape and I actually began to see it fold upon itself, that in itself is its 3D structure, and it's what gives the proteins its functional property. Some key pieces of the tertiary structure are that we have hydrophobic clustering. Typically, when the structure begins to take place, anything that's hydrophobic in the chain will actually cluster away from water properties. You'll typically see the hydrophilic polar amino acids toward the outside, and then you'll see ionic bonds and disulfide bridges hold together the structure. So a disulfide bridge is going to be where two sulfurs are chained together, and then you'll see an ionic bond helping to basically create this entire structure. And we'll look at that more in class. Our last level of protein structure is the quaternary structure, and this is typically where we see one or more of the subunits of the tertiary structure come together. So you can see, this is a really good example where you can see that here is, right here, two different subunits, the pink and the orange, to make the hemoglobin, and those four coming together, those are all the tertiary structures, but once you begin to put them together, you begin to make the protein complex, and in this case hemoglobin seen in your blood. Now it can go along, gather up the oxygen, and carry it throughout your body. So there are two different types of proteins. You have fibrous and globular. They sound like their names sound. So a fibrous one is fibrous, it's long and fiber-like, and the globular one is globby, it's just spherical. Fibrous proteins are typically for structural support, to give strength or structure, and the globular proteins are typically they typically have a function. So these may be your enzymes or antibodies. Fibrous proteins are typically insoluble in water, and while globular proteins are soluble. And a few examples for you. Collagen is a great example of a fibrous protein. It is a tissue, and keratin is your hair. Okay, this is it's even fibrous. It looks very fibrous. And then globular proteins, we gave two examples already. They do have functions. Amylase is an enzyme, breaks down your starch in your mouth, and then hemoglobin helps to carry oxygen. And then lastly, there is a significance between polar and nonpolar amino acids. So our nonpolar amino acids, these are the ones that do not have a charge and don't have an attraction to things that are polar. They are seen in membrane proteins, and they help to control the position of a protein within the membrane. So this picture is a great example. I like how it's color-coded and labeled, because you can see in this yellow portion right here that the nonpolar section actually helps maintain this position of this particular protein, because this yellow portion is attracted to the green right here, and this green portion is the lipid tail of our phospholipid bilayer, and so that is nonpolar. It is attracted to this nonpolar region of the protein and can actually help keep it in position so that the entire protein can do its job. It's bound to the plasma membrane. And our polar amino acids, they are also used within the membrane. So if you look, and I'm going to try to use blue, but right here in this red section, you've got the polar section right in the middle, and that's really convenient because things that are coming inside of this protein, you can see they have a positive charge. So if it has a charge to it, it's going to be attracted to that polar section. If there wasn't a charge to it, if we had flip-flopped the nonpolar and the polar regions of the protein, then those molecules that need to actually go through the protein would never go through, because they're not going to be attracted to them. And so what this ultimately is doing, it's creating a channel that is hydrophilic, it's water-loving. So let's say this is an aquaporin. An aquaporin would be used for it would be a protein that is going to channel water. Water can easily go through because water is polar, and so is the lining of that particular protein channel. And then lastly, polar amino acids will help in the specificity of the active site of an enzyme. And so we haven't talked about enzymes yet. They are a protein, but you can see in the red region we've got the polar site, and then the yellow region is all nonpolar, this part right here. So this part doesn't have any attraction for molecules, but this particular active site is the site where substrates will bind and a chemical reaction will take place. The enzyme will help catalyze the reaction, speed up the reaction, and so you can see that this particular red portion of the substrate is also polar. It's going to be attracted to the polar site, the active site, and then the reaction can take place. And so again, if you flip-flop those, the substrate would not have an attraction for the nonpolar site, and so you would actually not be able to speed up chemical reactions. So bring those notes to class. We're going to do some activities with proteins, we'll do some labs with proteins, and then we'll start to look more specifically at how these very specific proteins, enzymes, actually work.